all you have to do is look at pictures from the 1970s. Just look at the shapes of people, right? Fundamentally different. You did not see obesity, or if you did, it was extremely, extremely rare in the 1970s. Now, I don't know what the statistics are, 50+% of Americans are obese, right? That is purely environmental. Genetics have not changed in 50 years. So you probably don't know this, but I have a long history with resveratrol. Resveratrol, of course, is the miracle molecule in red wine. And I was one of the scientists who first debunked that whole longevity piece. Again, not saying there's no benefits to resveratrol. It is not a longevity drug. But even then, I rationalized drinking red wine. My sort of current speculation around NAD precursors is I think that boosting NAD for some people, if you have particularly, a high level of mitochondrial dysfunction or metabolic dysfunction, for those people, it may be beneficial. I think for the average person is probably not going to move the needle. There also may be some specific neurodegenerative diseases like Parkinson's disease where there's some intriguing clinical trial data.
Now, today's show with Matt Kaeberlein is loaded with amazing information. I strongly suggest that you look at the show notes and the timestamps, because towards the end, we talk a lot about rapamycin, metformin, the current state of testing and different tools to assess your biologic age and pace of aging. He also talks about and debunks some of the common myths about dietary supplements that have been purported to slow down your pace of biologic aging. We talk about other lifestyle factors such as socialization, exercise, nutrition, and more. It's a phenomenal show brought to you by Myoscience Nutrition. So as you know, exercise is one of the best interventions that you can implement right now to not only help improve your health right now, but slow down the pace of aging and optimize and prevent age-related chronic diseases such as obesity and diabetes and dementia and beyond. But one tool that can help your exercise sessions is the novel creatine-enhanced electrolytes by Myoscience. What makes this product unique and different from many of the other creatine products out there or the isolated electrolyte products out there is this pairs the best of both electrolytes and creatine together. Creatine helps with cellular hydration, and electrolytes help enhance the absorption and utilization of creatine. So they work synergistically, and they can help improve your exercise efficiency and exercise sessions. There's close to 900 reviews over at myoscience.com from people just like you who are finding benefit from the novel creatine-enhanced electrolyte sticks. I strongly suggest you give these a try. There's a 30-day money-back guarantee over at myoscience.com. I'll put links in the description below. Most people find that they get the most benefit from using this before or during their exercise sessions. There's a lot of great customer feedback and reviews that you can check out, and you can take advantage of the fact that there's a 30-day money-back guarantee. They want you to get great results from the formulas that they produce. So use the code podcast to save over at myoscience.com. So let's cut back to it with Matt. Matt Kaeberlein, thanks for coming back.
Absolutely.
How would you explain to people what you do in terms of in the realm of longevity and studying the biologic processes of aging?
Yeah, so I guess I would say by training, I'm a scientist. So I spent about 25 years actually doing basic research on the biology of aging and what are the mechanisms that connect the aging process with functional declines and diseases. And then about a year and a half ago, I left my academic position. I still have one foot in the academic world, but now I am CEO of a company called Optispan, which is really at the far end of the other end of the spectrum when it comes to human health, which is really all about trying to create tools and technologies to enable health span optimization for as many people as possible.
I love that. And what are your go-to tools and technologies? I know you do functional ththings like DEXA and VO2 max, and how esoteric do you get with that?
Yeah. So, I think we start with the fundamentals. So, at Optispan, we talk about the pillars of health. I think, lots of people have their pillars of health. We like 4 because that's a nice, easy number. And we like action words. So we look at eat, sleep, move, connect as the foundation. And those are really what you would think they are. Eat is nutrition, move is exercise, sleep is sleep. And connect really refers to both your individual connection with the world around you. That would include things like mental health, and also your human connection. Both of those things we know are super important for quality of life. And we think that that's the foundation. And so one of the first things we try to do is at an individual level, identify, which pillar is weakest and can we take action there. Of course, we also do a very medical approach to health span. So for our clients, we have what we call a gateway day. And that's the day where we really try to understand at a deeper level where each person is at on their own health span trajectory. So we use things like comprehensive blood work, imaging, so ultrasound, DEXA to look at body composition, assessments for physiological function like VO2 max and strength. And then of course, part of this is also about making sure there isn't pre-existing problems that we need to take care of. So we do things like advanced cancer screening or assessments for different age-related diseases, all with the goal of, again, figuring out where each individual is at and then trying to identify, where's the biggest bang for the buck going forward for how we can start to modify either lifestyle or, when appropriate, medications to get people onto a better health span trajectory.
Amazing. You mentioned connection, and I think that's really important, but yet under-recognized, especially within the mainstream medical community. I know you're going to Brazil next week and have presumably traveled throughout Europe. It's amazing to me when you look at different parts of the world, the diet doesn't appear to be that, obviously maybe a little bit less processed foods depending upon where you are, but generally I've found higher prevalence of smoking, for example, in Europe.
Yeah.
But yet people are walking more and connecting more. So how much do you think that plays into the social aspect and connecting and movement? How much of a lever when it comes to longevity do you think that pulls? Yeah,
it's a good question, and it's hard to answer. So I'm a scientist, so I'm always like, can I answer this quantitatively? What are the error bars, right? But I think from a sort of more pragmatic perspective, for longevity, we know that human connection plays a role. I don't think we know how big of a role. So my guess is, that may be less important from a pure lifespan perspective than things like what you eat and how much you eat and how much you exercise. But from a quality of life perspective, it's huge. And like you said, I think massively underappreciated. And I think, again, this is a generalization for sure, but I think particularly for men, and I'm 53, in my sort of demographic, I think it is even maybe more underappreciated. You hear about this epidemic of male loneliness. Mm-hmm. Again, I'm not at all trying to say that connection isn't a challenge for some women as well. But I think for men in particular, this is an area that we tend to neglect, which is hugely important. And when I look at my pillars, right, again, I try to practice what I preach. Eat, I've gotten a lot better. Move, I've been pretty good throughout my life. I've gotten a lot better. Sleep, fortunately, I've never had a problem with. Connect is the big whopping, this is where your weakest link is for me. Mm-hmm. And so I'm trying to take steps to fortify that. And I think there are lots of ways that you can do that. And again, I sort of grouped both your personal connection to the world around you, your mindfulness, along with human connections and relationships. I think I'm weak in both, but I think, one of the things that I've started trying to do is to actually focus a lot more effort on not just connecting with other people, but rebuilding and strengthening relationships that I maybe haven't been very good at keeping up with as I've, gone through my life. And so I totally agree. I think it's hugely important. I don't know how much that contributes to differences in health span in different parts of the world. Certainly, the United States culturally is less adept at maintaining and nurturing relationships as we get older compared to other parts of the world. I think particularly family relationships, extended family. I think, many people are very good and focus a lot on their nuclear family, marriage, kids. We really don't have the same sort of extended family connections in general in the United States that you see in other parts of the world. And I think that is clearly important for quality of life and could have a big impact on differences around the world.
Right. And it's really hard for people to, it's very easy to eat better and to exercise more.
Well, I don't know that I would say that because a lot of people struggle with it, but I think you know what to do maybe.
Exactly.
Yeah.
Like it's harder to go out and make friends, relatively speaking, compared to changing your diet and exercising more. That's where I was going with that. So
yeah, I mean, I don't know. Yeah. So I think it seems harder. I think there are lots of things that almost anybody can do. Like, I agree. You can't just walk out the door, find some random person and say, hey, can I be your friend? I mean, you could. You probably wouldn't get the reaction that you're going for, but you might. You never know. It would be an interesting experiment. But you certainly can take up new activities with a group, right? So like one thing I still need to do at some point, right, that I've talked about for years is I miss the sort of competitive team sports, right, that I used to do when I was younger. And I've talked forever. I played softball a little bit when I was at the University of Washington, but I've talked forever about why don't I should join a softball league, right? That's the kind of thing. You could go join a softball league. You could join a hiking club, right? And there you're actually ticking off two pillars, move and connect, right? Or, any hobby, maybe a hobby you don't have that you might like. Go join a book club or a sketch club. There's all sorts of stuff you can do where you can interact with people and do something else that also brings you joy. And again, I feel almost like I shouldn't be talking about this because I'm trying to figure it out for myself. But I think, we sometimes make this seem harder than it really needs to be. It's really about making a commitment. And so, one of the things that I think is a good way to approach this is to just take a look at your pillars, identify the pillar you're weakest in, and then really find one thing. What is one thing that I can do to strengthen that pillar, and then make a commitment to do it and do it for a month, right? And see if you can make a new habit. Because I think a lot of this comes down to new habits, and then reevaluate, and maybe you find a different pillar that's now the weak link.
Right. Now, once you prioritize one, move on to the next and be goal-oriented. I love that. Speaking of one pillar of aging and one mechanistic aspect of aging, we talked in the sauna before we did. How do you feel after that, by the way?
I feel awesome.
Yeah, amazing. Yeah, your brain is you're on fire, which is great. So we were talking about mTOR, because if you look at the research on aging, especially when it comes to diabetes and obesity related conditions, it seems that mTOR is this key pathway that's overexpressed and can cause cells to misbehave and age more rapidly. So that's one end of the spectrum. On the other end, we know that we should preserve strength span or strength as we get older, preventing muscle loss and sarcopenia. So I think there's a lot of confusion for people when it comes to, these are sort of competing ideas, right? On the one hand, we want to periodically express mTOR, prevent muscle loss. On the other hand, we know that potentially overexpressing this mechanistic target of rapamycin could be problematic. How do you reconcile this and help people navigate the waters of protein and resistance training?
Yeah, I mean, it's really complicated. I think there's a couple things to just sort of level set, right? So one is we know that mTOR is this fundamental gatekeeper of growth. Really what mTOR does is it senses the environment around us and helps the cell or the organism, based on the environment, make a decision. Should I grow or should I shut down growth and become stress resistant? And so what we've learned from studies in laboratory animals is that if you turn down mTOR signaling, you can slow aging and increase lifespan. I think it's important to say we don't know for sure that the same thing is going to be true in people yet. I think it probably is to some extent, but we don't actually have that data. But it's also important, and I kind of said this, to recognize mTOR is not an on-off switch. Think of it more like a dial that you can turn up or turn down. It's also not necessarily turned up or down to the same level in every tissue and organ. So you talked about muscle function. You're talking specifically about mTOR signaling in the muscle. Whereas we also have mTOR in our brain, and it's important for different functions in the brain. We have mTOR in our liver, our kidney, our heart. So the optimal level of mTOR signaling for health and function may be different in different tissues and organs. And then the third thing I would say is the idea that mTOR becomes hyperactivated with age, I would say is still a little bit unclear. I would say we can be pretty confident that mTOR activity is not optimized for longevity, but being optimized for longevity is not necessarily the same as being optimized for function, or certainly not the same as being optimized for reproductive success, which is really what evolution is driving us toward. So all of those are sort of caveats to say that where I land is that mTOR needs to be turned up and turned down in an appropriate way. And you can accomplish both of those things by interventions like eating protein or exercise, with both of which will turn up mTOR, at least transiently, or turning it down by rapamycin. I think the question that people get stuck on is, if I take rapamycin or if I eat a low protein diet, will I be unable to gain muscle, or will I lose muscle? And will that become detrimental as I get older? Certainly, I think I'm pretty confident saying that you can take rapamycin and not lose muscle. I know this because I've experienced it, and I know lots of other people who have. If you took enough rapamycin, would you lose muscle? Probably. But again, this is about turning the dial as opposed to turning a switch on or off. What I can't say with any level of certainty is that if you take rapamycin, say once a week, which is what most people are doing, and you do resistance training, that you're going to gain the same amount of muscle that you would gain if you don't take rapamycin. You can still gain muscle, but you might gain a little bit more if you weren't taking rapamycin. I just don't think we know. There are clinical trials, and we can talk about the clinical trials. There are clinical trials that are starting to address that question. But from my perspective, I don't think it really matters, because at least for most of us, when you get in your 50s, 60s, it's not about how much muscle can I gain. You're not, most of us, trying to be professional bodybuilders. It's about how can I best maintain function as I get older, so that when I get in my 70s, 80s, 90s, hopefully hundreds, I can do what I want to do, or at least do most of what I want to do. And so that's where I think you have to really think about this cost benefit. And I really don't see any evidence in the literature that rapamycin is detrimental for lean mass. If anything, in the animal studies, it preserves lean mass. So mice and rats taking rapamycin are protected against age-related sarcopenia. But yes, it might be that if you're trying to be a competitive powerlifter or bodybuilder, you're not going to get quite the same gains you would taking rapamycin. Protein, I think, is a little bit of a different beast, right? So, and one way to think about this is rapamycin is a very, very specific drug. It has one biochemical target and one only that we know of. Protein, on the other hand, is a very, very dirty biochemical drug. It has thousands of different biological reactions that are impacted by dietary protein. So I don't know what the answer is in terms of optimal protein levels. My reading of the current sort of state of the literature for humans is that a higher protein diet in general is better for preserving muscle function and muscle mass as we get older, as long as you are also doing resistance training. I think a lot of times the confusion around the risks associated with a high protein diet come from studies where they look at people who are eating a typical American diet, probably obese, probably not exercising regularly. I think in that context, it's clear that a high protein diet does at least somewhat increase cancer risk and potentially metabolic risk. But in people who are eating a relatively clean diet, exercising regularly, not overweight, I think the bulk of the data support the idea that a higher protein diet is probably better than a low protein diet. What does higher mean? It's all relative. I don't know that we have great data on this, but I think the rule of thumb of 1 gram per pound of body weight towards the upper end, but being somewhere near that, is probably about right. And that's what I try to do.
Awesome. And when it comes to protein sources, are you worried about the saturated fat and cholesterol in red meat versus lean chicken? Where do you stand with that?
I do think it's like anything else. I'm not a guy who says don't ever eat red meat. I like red meat. I eat red meat periodically, but I think it's like anything else. Moderation is important, and certainly I think that and again, I'm going to probably take heat from this from my wife, but I think the negative reputation of red meat has been a bit overblown. Having said that, I think if you only eat red meat, that's probably not a great strategy for health. So I think if you want to have it once in a while, great. I eat a lot of chicken, a lot of fish. Both are great sources of protein, probably healthier than red meat. But I think kind of similar to the case that I laid out for high protein, I think a lot of the epidemiology on red meat suggesting that there are significant health risks associated with red meat come from studies where most of the people eating a high amount of red meat were also eating a pretty crappy diet, probably overweight, probably not exercising regularly. So I think overall context is important when you're trying to look at the impact of one macronutrient or one food type. You kind of have to look at the sort of overall health status of that population. And it could be very different in different people who have fundamentally different lifestyles.
Right. Really good point. Going back to the rapamycin, I think there's a lot of interest in this as of late. What is a lot of people are like microdosing this 2 to 6 milligrams, like you said, once or twice a week. What are some of the clinical trials looking at now in terms of dosing, frequency, and then the target population?
Yeah. So there are at least 3 clinical trials that I think are really interesting right now in this space and asking very interesting questions. So one is related to muscle function, which we already talked about. Actually, there are a couple going on. Where the one that I have sort of helped just advise on design is a clinical trial out of New Zealand where they're looking at the impact of rapamycin in older adults also practicing resistance exercise. And so, it's a small trial. That's one thing that's worth saying. All of these trials are underpowered for efficacy, unfortunately. We could talk about that. That's a bigger problem in this entire space because the funding for research around longevity interventions still is pretty small. What you see are a lot of underpowered clinical trials where really they're safety trials and they hope they get a hint of efficacy. So that's just important to kind of set people's expectations. But even still, I think there's an opportunity here to see whether or not rapamycin has any negative impact on either strength or muscle mass, body composition changes. There will be a clinical trial reporting soon. It's the Pearl trial that looked at body composition with people taking rapamycin by DEXA. And so that will also be interesting to see how that comes out. So that's one area. Another is around periodontal disease. So my lab at the University of Washington showed that in mice, first of all, mice, as they're aging, normally develop periodontal disease. So they lose bone around the teeth, they develop inflammation of the gums, gingivitis, and they show pathogenic remodeling of the oral microbiome, just with normal aging. And if we give old mice, 10 weeks of rapamycin, we can reverse all 3 of those things. Okay. So now there's a clinical trial by John Ahn, who he was the graduate student who did that work in my lab. He's now a professor of oral health sciences at the University of Washington. He's doing a clinical trial to look at whether or not rapamycin in people with periodontal disease can have a positive impact on those 3 clinical endpoints. So that'll be super interesting. I think periodontal disease is underappreciated, much like human connection is underappreciated for its role in total overall health span, or oral health in general, I should say, not just periodontal disease. Underappreciated how big of a role that can have in health and potentially longevity. So that'll be interesting to see if rapamycin has an impact there. And then the third area, which is very different, is looking at reproductive aging in women, and particular premature ovarian senescence. So again, we know from studies in mice that rapamycin treatment can delay ovarian senescence and, at least in some cases, partially reverse ovarian senescence. So you can actually see a reversal of the atrophy of the ovaries in aged mice. So Yushin Su and Zev Williams at Columbia University are doing a clinical trial, enrolling now, to look at whether or not in women with premature ovarian senescence, rapamycin treatment can preserve ovarian function and perhaps boost fertility in those women.
That is incredibly fascinating. I'm sure you've seen the videos. I mean, there's a woman in the UK who was going through what she dubbed menopause, but it was just premature ovarian failure at 18 years old. There was another actress or musician. I mean, in the last year, this has really become like a sensation phenomenon, if you will, unfortunately. And I think AMH levels are dropping and all these sorts of things. So that to me is incredibly practical, because if you look at fertility rates in both men and women, they're declining.
And it's also, I think, important to consider, like, not only what is the connection between ovarian senescence and biological aging, which is super important, interesting biology. In fact, this afternoon, I'm going to be interviewing Jennifer Garrison from the Buck Institute, who's one of the leading people on reproductive health in women, who does research in this area. So I think that's super interesting. But also, why is it the case that this seems to be happening in more people at an earlier age? And what are the environmental or lifestyle factors that are potentially causing earlier ovarian senescence, earlier entry into menopause for many women? And related to that, it's worth, I think, just stating explicitly that I think it's pretty clear that the 4 pillars we've already talked about influence biological aging, and they probably influence onset and progression of menopause in women. So lifestyle can play a big role here. I certainly wouldn't want to suggest that, rapamycin is going to be the cure. I think, for people who are thinking about what can I do in my own life, I think it's clear that a healthy lifestyle can have a big impact, not just on your sort of overall biological aging process, but if you're a woman, on your ovarian aging process.
I mean, it's just amazing to me that these conditions we didn't hear about even 10 years ago. All of a sudden, it seems like in the last several years, I mean, not only this premature ovarian failure, but early onset cancer. I mean, that was not a term that was in the literature. And now in the last 2 years, we're hearing all about that. So it seems that something in the environment in the last 15, 20 years has significantly changed. And, the rate of biologic aging, which is affecting the function of these organs, is causing things to just accelerate at a greater level. I mean,
I don't know. I think you could certainly you can speculate that's the case. I'm a little bit more cautious because you always have to wonder like how much of this is more reporting and given, how concepts can go viral without a lot of data on the internet. So I don't know yet. In part, I haven't done a lot of digging to really try to look carefully at the rates. I certainly know there is a lot of talk of increased cancer rates, at least for certain types of cancer in younger people these days. I think that's probably real. I think it probably is environmentally driven. But I also think that shouldn't be too surprising in a sense, because if you just look at how our environment has impacted health going back 50 years, it's enormous. I mean, again, all you have to do is look at pictures from the 1970s. Just look at the shapes of people, right? Fundamentally different. You did not see obesity, or if you did, it was extremely, extremely rare in the 1970s. Now, I don't know what the statistics are, 50+% of Americans are obese, right? That is purely environmental. Genetics have not changed in 50 years. And so it shouldn't be surprising that as our environment continues to change, there are going to be impacts on health. And I wouldn't want to suggest they're all negative. I think there can be some positive impacts on health from environmental changes as well. I think the key and what would be great is if we could get ahead of some of these things instead of being reactive like we were with obesity, then maybe we could actually prevent some of the more detrimental effects of these environmental changes.
Yeah, I agree 100%. If we could close the loop on the optimal dosing. And I know this is sort of off label as a preventative prophylactic for rapamycin. Where does, if there is any research to suggest what from a prevention standpoint, everyone is working on the 4 pillars that we talked about, fasting, exercise, connection, and all that.
We didn't talk about fasting.
Well, I think it's implied. The time-restricted feeding, I mean, what do you think about this research?
I'm completely unconvinced,
really?
To be honest with you. So here's what I would say. I think fasting and time-restricted feeding or eating, depending on whether you're a mouse or a human, clearly can have benefits if it's in the context of overall caloric consumption. I think for a lot of people, those are useful strategies for maintaining a healthy body weight and not overeating. I think for longevity, lifespan, the data are almost non-existent that fasting or time-restricted feeding in laboratory animals increases longevity independent of calories. So if it's also caloric restriction, it will extend lifespan. There's no evidence really that it has an effect on longevity outside of that. In people, I think it's mixed, but people are hard, right? Again, trying to really do these studies well in humans is challenging. There are certainly biologically plausible mechanisms by which fasting or time-restricted eating could have beneficial effects. I think if done in certainly time-restricted eating, if done in a way that matches your circadian rhythm, can strengthen circadian rhythms in some people, maybe improve sleep quality. So I think for some individuals, but as a pure longevity intervention, independent of caloric restriction, I just don't think there's any data to back that up.
It seems like people get really excited about it, though, on the internet, fasting.
Definitely people get excited about it. And I think, again, I'm not saying that there's no benefits, and certainly for some people there are. I'm coming from the angle of, is it impacting the biology of aging in a way that will slow aging and impact lifespan or health span? If it's not also caloric restriction, there's no real evidence to support that. Now, you could certainly make the case that if you were, let's say, fasting, I don't know, once every 3 days, that's pretty extreme, but let's say you did that. The likelihood that in the 2 days you're not fasting, you're going to make up for that day that you're fasting is pretty low. If you did, I don't think there's much reason to believe it would be beneficial for you. So I think it's more about context, right? And again, I think fasting or time-restricted eating can be a very useful and easy way for some people to also limit their total caloric consumption. The last thing I'll say on this, though, is I think it's a total mistake to think that fasting or time-restricted eating, if you don't also pay attention to what you're eating, is going to have any big benefits for you. I think paying attention to the quality of your diet is probably, at least in my view, more important than trying things like fasting or time-restricted eating.
Great perspective. What do you make of the Latin American and European lifestyle of having dinner at 11 p.m., right? Because you mentioned the obesity rates here in the U.S. are north of 50% in certain parts of the U.S. You go to, like, you're going to go to Brazil here in a little bit. I was in Latin America, last year. People start to go out at 10:30, 11 o'clock at night, and they eat dinner late, and they're not really, to my knowledge, sleeping into noon. They're getting up and having breakfast again. So for all the talk about compressing your feeding window, it seems like there's less obesity. The top causes of death are completely different there in terms of heart disease, I think, is at 2 or 3. Yeah. Alzheimer's is 1, at least in Colombia I looked at. So what do you make of that?
So again, I think it's really hard to draw strong conclusions. I think you can, and in part you also have to look at what is the sort of distribution of life expectancies, right? Because that's going to have an impact on frequency of different age-related diseases, things like that. But I would say, I think what we can probably conclude with some degree of confidence is that these things about like the window that you eat in are not going to be major determinants of population longevity, right? And it's going to be the other factors like social connection, which is, again, you could certainly speculate going to be greater in countries like that, or activity level. I think also, and this is where it's a little bit hard to get really rock solid quantitative data around diet quality. My speculation is diet quality is probably worse in the United States than just about anywhere else in the world. How much worse, I don't know, but it's probably worse. And I think diet quality probably is contributing to all of these things. Sedentary lifestyle in particular, though, is probably the big difference. I am shocked at how many people in America, I mean, when we say sedentary, I think we probably don't think how sedentary a lot of people really are. And I think that has a huge impact.
Yeah. I mean, we're talking, they don't leave the house. Everything's being delivered. Not even getting out of bed or washing themselves. Yeah. Wild stuff. What's interesting about Europe and other parts of Latin America, the smoking prevalence is much higher.
So one thing I say sometimes about smoking is, I mean, so smoking and obesity will both shorten lifespan. Smoking will kill you fast. It has a much lower impact on health span than something like obesity. So smoking will kill you fast. Obesity, diabetes will kill you slow. They both will negatively impact lifespan.
Yeah. So just don't smoke. What do you think about vaping? A lot of people, my barber was telling me how she quit smoking, but now she vapes.
Yeah. I don't have a strong opinion other than it's probably not a great idea. Is it better than smoking? Probably. But again, if your goal is to, get as close as you can to optimal quality of life, optimal health span, very likely there are going to be significant negative effects associated with vaping. Mm-hmm. I don't think we know yet exactly how big the negative effects are going to be, and I think it's probably the case it's not as bad as smoking. But I totally get that addiction, whether it's behavioral, whether it's chemical, is real, and so, if people are addicted, it's probably a better strategy than smoking. But to the extent you can, maybe that's the pillar to work on if that's something you're struggling with.
Yeah. What about alcohol? Where do you sit with that?
Yeah. So, I mean, personally, I've dramatically reduced my alcohol consumption in the past 3 years, and I think it's been one of the best things I've done for my health. So, mentally, physically, metabolically, it's had a huge impact. And I would say, of the health changes that I sort of have gradually, gotten more dialed in, that was one of the later ones that I made, but it's had a huge impact. So I think what we know about alcohol is that it definitely has a negative impact on sleep quality. I don't think anyone can debate that. The epidemiology is complicated. So you can make a reasonably good case from epidemiology that one drink a day results in lower all-cause mortality, so lower risk of death than no drinks a day. There are all sorts of confounding factors that could account for that. Is it the alcohol or is it some other reason? I don't think we know. But there are lots and lots of studies that show that. I think it's important to appreciate those are like one European drink, not one American drink. And it's very hard for a lot of people to stop it at one drink. So, my view is if you like to have a glass of wine with dinner, no problem. If you like to go out with friends once in a while and do it in the context of a social sort of environment, that can have some benefits independent of the alcohol. I don't think anybody should be drinking alcohol or particularly red wine because they think it's a longevity intervention. I think the net risks are way higher than potential rewards for most people.
Yeah, well, I agree with you with regards to alcohol. I've significantly cut down my alcohol consumption and have noticed just sleep quality, overall energy, mental clarity to be significantly improved. Yeah. And I was one of those people that would rationalize consuming red wine to say that it's heart healthy or the polyphenol content is good for me or
yeah. So you probably don't know this, but I have a long history with resveratrol. So resveratrol, of course, is the miracle molecule in red wine. And I was one of the scientists who first debunked that whole longevity piece. Again, not saying there's no benefits to resveratrol. It is not a longevity drug. But even then, I rationalized drinking red wine. So I think there's all sorts of mental tricks we can play with ourselves to do things that we want to do that maybe aren't best for our health. But
and you said it's not a longevity supporter. All the research on sirtuins and that from Sinclair's lab.
It's pretty complicated, but here's what I would say. Whether or not resveratrol can activate sirtuins in vivo, by that I mean in cells in our bodies, is unclear. The original report was a test tube artifact. So I can't rule out the possibility that some context, some specific target, resveratrol activates sirtuins. But I think the preponderance of evidence and certainly an unbiased view would suggest that it probably doesn't to any great extent. More than that, though, there was a meta-analysis published last year, I think, where they looked at least as many of the published longevity experiments with resveratrol across all organisms and asked what was the sort of median effect. 0.
Wow.
0. And that's with a negative publication bias, meaning people usually don't publish results that don't work or where there's a negative effect on lifespan. So probably, if anything, in the preclinical literature and laboratory animals, resveratrol has 0 or a depressing effect on lifespan. Wow. So again, there's all sorts of literature out there suggesting that in certain contexts, at very high doses or whatever, that resveratrol may have some positive beneficial effects on a variety of different conditions. I'm not ruling that out. But I think, again, if you want to look at it through the lens of longevity, it's very, very hard to make a case that resveratrol, there's any rationale for resveratrol as a longevity drug.
Now, what about spermidine? That's been really popular as of late.
Spermidine is pretty interesting. So spermidine, is a natural component of our diets. Different people get different amounts of polyamines. It's a class of molecule for polyamines. Seems to be an autophagy booster. That data seems pretty clear, at least in cells. Again, the extent to which spermidine acts to boost autophagy in tissues and organisms of animals, I think the data is a little bit less clear there, but it seems to be an autophagy booster in cells. I think the lifespan data, at least up through invertebrates, is pretty convincing, and there is some, not a huge amount, some longevity data in mice and some epidemiology, so correlational associations with polyamine intake and longevity in people. So I kind of put it in that class of sort of second tier longevity interventions that seem interesting. There's some smoke there, probably some fire. To what extent just in taking spermidine as a supplement, not in the context of a normal diet, to what extent would that have a positive effect on autophagy or longevity is unclear at this point. But, it's intriguing and worth paying attention to for sure.
Are there any other compounds out there that are popular right now? I mean, it seems like every year there's some new compound that's purportedly some superfood and so forth. Since Resveratrol came on the scene in 2006, anything like that actually excites you?
Yeah, I mean, I think there are some things that are worth paying attention to, right? So, and again, I kind of put these in two buckets. There are pharmaceuticals, right, where you would need a prescription, or there are natural products that potentially are being sold as supplements you get over the counter. It's harder for me to get excited about a supplement, mostly because you rarely, if ever, get the quality clinical trial data that you will sometimes get with pharmaceuticals, right? So I think the supplements people are excited about that, again, I might put in that sort of, second tier group. Alpha ketogglutarate's interesting. Did not reproduce the lifespan effect in mice. Recently, that was published by the Interventions Testing Program. But I think there's enough health span data to still kind of keep it in that second tier category. Urolithin A is interesting. Also not yet much human data, but a couple of small clinical trials. So urolithin A is thought to be a mitophagy booster, so autophagy targeted towards mitochondria. Probably does other stuff for mitochondrial health as well. So I think that's pretty interesting to think about. NAD precursors are the one that people are really, really excited about still. Mixed data. So there's a lot of smoke with the NAD precursors. There's also a lot of really bad science in that space. And so it's hard for me to have a ton of confidence at this point. My sort of current speculation around NAD precursors is I think that boosting NAD for some people, if you have particularly, a high level of mitochondrial dysfunction or metabolic dysfunction, for those people, it may be beneficial. I think for the average person is probably not going to move the needle. There also may be some specific neurodegenerative diseases like Parkinson's disease where there's some intriguing clinical trial data. The other thing to say about NAD precursors, again, because I know probably a lot of people listening have heard of NAD precursors or maybe taking them. Yeah. So taking NAD won't get into your system orally. You can do an NAD drip. I don't have an opinion on NAD drips. I've never done one, but taking it orally won't get into your system. You can pay for nicotinamide mononucleotide, which technically the people selling it should not be selling it, but they still are. And nicotinamide riboside. Both of those, I think, will boost NAD levels in the liver, may boost NAD levels in some other tissues and organs. But you could also take nicotinamide or niacin and do the same thing. And it's pretty clear that most, maybe all of the NMN and NR is broken down by the gut microbiome to niacin or nicotinamide and gets taken up that way. Wow. NR might also get taken up directly by the gut. I just interviewed Charlie Brenner, who's the person who discovered NR. And he's very much of the opinion that it is bioavailable, but most of it is probably being broken down before it gets taken back up and resynthesized into NR and NMN. And then if all goes well, back to NAD. Last thing I'll say on this, there was a study that just came out. Actually, I'm not sure it's been peer reviewed yet. It's a preprint. In aged mice, where they treated the mice with NMN at doses that had been shown to have positive effects on health in brain and kidney. In that study, brain and liver, not kidney. In that study, they were actually looking originally at heart function. They saw positive effects on heart function in aged mice, but then they also looked at the kidney and they saw pathology in the kidney. And they make a pretty compelling argument that super physiological doses of NAD in the liver get metabolized to byproducts of NAD. And they listed a few. They focused on one called 1-methylnicotinamide that the kidney has to clear. And in a young kidney, that's no problem, can handle it just fine. In an aged kidney or in a diseased kidney, it might actually push things over into the realm of pathology. And I don't know if anybody has ever looked at kidney function, kidney pathology in the context of aging and NAD precursors. I'm not like raising major alarm bells, but I think it's enough it's concerning enough that people should really look at this and get this figured out. It could be the case that at least high doses of NAD precursors could cause some problems in at least some people who are prone to kidney dysfunction.
Interesting.
So those are the ones that come immediately to top of mind. And then on the prescription medication side, there's just a couple things to say. So there are a few that have come out of the interventions testing program in mice as increasing lifespan. Acarbose is one. That's an anti-diabetic drug that prevents breakdown of complex sugars into glucose, and so reduces blood glucose by reducing uptake. Similarly, SGLT2 inhibitors, canagliflozin in particular, increases lifespan in mice. That reduces blood glucose sort of at the other end of the spectrum by preventing reuptake by the kidney, so you pee more of it out. And then the other one that is pretty compelling is estrogens. So both 17-alpha estradiol and estriol have been shown to increase lifespan in mice. Now, the reason why I think those in particular are really interesting, especially the SGLT2 inhibitors and the estrogens, are in a recent study from UK Biobank, where the researchers looked at essentially all of the prescriptions that had over, I don't know, it was 500 or 1,000 in their data set, and then tried to match them to people of similar health status and look at all-cause mortality. Most prescriptions were associated with higher mortality, so increased risk of death. That's what you would expect. There were 16, I think, that were associated with lower all-cause mortality. Like 6 of those were estrogens, which is amazing. There's super interesting biology there. Going back to what we talked about earlier about ovarian failure and menopause, really, really interesting biology there. Interestingly, in the mice, 17-alpha estradiol works better in male mice than in female mice. So we could spend a whole show on this, but I think there's something cool there. And then the SGLT2 inhibitors also came out. Multiple SGLT2 inhibitors came out as being associated with lower all-cause mortality when compared to people of similar health status. It's mostly going to be probably diabetics who are taking these things. Interestingly, metformin did not come out as having reduced all-cause mortality. And this kind of fits with, what some of us have been saying for years, which is that the pro-longevity evidence for metformin was always pretty weak. So the original mouse study showing lifespan extension from metformin was in a very short-lived strain of mice that all died from a specific type of cancer. When it was tested in longer-lived strains of mice, it either had a tiny, tiny effect, positive effect on lifespan, or had no effect on lifespan. And it sort of got a lot of attention for potentially being beneficial for human longevity from a study that initially reported certainly that diabetics taking metformin live longer than diabetics who didn't take metformin, who took other drugs. And they made a hint that diabetics taking metformin might live longer than non-diabetics not taking metformin. If true, that's really, really compelling. Turns out that hasn't been reproducible in 2 subsequent studies. So I think the perception of metformin as a longevity drug has shifted in the last few years. Not as much enthusiasm there. Certainly it's a really, really solid anti-diabetes drug, may have some beneficial effects, particularly in diabetics for other age-related diseases, heart disease, dementia, some forms of cancer. But in non-diabetics, I don't think there's a lot of evidence for benefits from metformin at this point, and some concerns around offsetting some of the potential benefits of exercise. So again, risk reward, I personally don't put metformin in the reward is favoring risk at this point.
Was there enough people on rapamycin in that data set?
No, it did not make their cutoff for enough people taking it, mostly because still it's a pretty rarely prescribed medication, mostly in organ transplant patients, some rare forms of cancer, but not a lot of people on the drug.
Yeah. Going back to urolithin A and mitophagy, if people are exercising, which is arguably one of the best ways, would that be a worthwhile supplement, or is that kind of?
I don't know. Again, I don't know of any data. I should be careful because I feel like there might have been one small clinical trial where they looked at muscle function. Certainly there's some evidence in people of improved mitochondrial markers. They may have looked at muscle function in that trial. I can't remember if those people were exercising or not. My intuition is that, you're going to get big benefits from exercise, particularly if you're not trained, right? And that if urolithin A or NAD precursors or anything else or rapamycin have positive effects on top of that, they're going to be relatively small compared to the benefit you get from just practicing resistance exercise.
Mm-hmm. And you mentioned the NAD precursors. To close the loop there, so just taking good old niacin is probably, that's a fraction of the cost. I mean,
it's like a 10 to 50 fold cheaper. Yeah.
Yeah. I mean, people have been using that for...
Yeah, I mean, some people flush on niacin, so it does have that side effect. But in terms of its ability to boost NAD, yes, you should be able to get the same effects on NAD from niacin that you get from nicotinamide riboside or nicotinamide mononucleotide. That still leaves the question of, is boosting NAD net beneficial for most people, which I don't think we know.
Mm. And just for folks that don't understand, why would they consider potentially boosting NAD?
Yeah.
What's the value?
Yeah, so there's some evidence. This I think is an interesting case, much like resveratrol, maybe not quite to the same extent as resveratrol, where the sort of perception or dogma in the field got way ahead of the data. Yeah. So there is this sort of perception that NAD levels universally decline with age, and that leads to impaired metabolic function and mitochondrial function, because NAD is a cofactor that's required for hundreds, if not thousands, of metabolic reactions. And there is also the sort of surrogates of NAD, NADP and NADPH, which are also super important for biosynthesis and all sorts of metabolic reactions. So really important molecule. But it got popular in the longevity field because sirtuins, which at the time a lot of people thought were potent longevity targets. They probably aren't, but a lot of people thought they were. Because sirtuins use NAD, they consume NAD, there was a model that became popular that if you could increase NAD levels, you could activate sirtuins, and that would be beneficial for health and longevity. So you had the two sort of arrows, one, NAD levels declining with age, two, boosting NAD, activating sirtuins, that suggested that if we could sort of artificially, therapeutically increase NAD levels, that would have beneficial effects. Since then, we've learned there are other things happening that also deplete NAD, like DNA damage or hyperactivation of the immune system. So I think there's good reason to believe that at least in certain tissues, or at least in certain contexts, like you get exposed to COVID-19, you have a severe immune reaction, NAD levels will at least be transiently depleted. And that will have an impact on fundamental biology, metabolic function. So boosting NAD in those contexts probably is a good thing. And so I think there's some reason to support that. But the idea was that if you could turn up NAD, you would slow aging. And there were some initial studies that seemed to support that. So a study in yeast suggested you could increase lifespan in yeast, a study in C. elegans. So it kind of hit the invertebrate target. And then there was a paper published out of Johan Auwerx's lab in mice, where they started treating mice in middle age and reported an increase in lifespan and improvements in some health span metrics. So that result has not been reproducible. I'm not saying it's wrong. The studies were done a little bit differently, but it has not been generally reproducible that you can increase lifespan, or I would say even generally increase health span in mice from NAD precursors. And so at this point, I feel like, there's a body of literature reporting benefits from NAD precursors. There's a body of literature reporting no benefits from NAD precursors. And there's probably another body of literature that, again, as we talked about earlier, never got published because people don't publish negative results, also not showing any benefits in the context they were looking at it from NAD precursors. So on the preclinical side, it's kind of hard to know where to land. On the human side, there have been a series of small clinical trials, none of which have shown, at least to my knowledge, any concerning outcomes, like really bad side effects. Also, they typically haven't hit their primary endpoint. So they were underpowered, but they didn't typically hit their primary endpoint. So I would also put those in the category of maybe. So I think we just don't know. That's kind of where I'm at with the NAD precursors, maybe.
So I'm not super excited about it. You'd focus more on VO2 max or strength or other proxies of health span?
Yeah. Well, so in terms of what you measure, right, here's the way I think those are two different things. I think if you took NAD precursors and you did a study and you showed that, somebody's body composition went in the right direction and their VO2 max got better, or their strength got better, I'd be like, yeah, that's a meaningful outcome. If you show that you take NAD precursors and you raise NAD in your blood, I'd be like, that's what we'd expect. Does that mean anything? I don't know. So I think what you measure and what the endpoints are is important. And I tend to gravitate towards functional outcomes as much as possible because I feel like we don't have a ton of biomarkers we're super confident in, at least in the longevity space. This might be a good time just to quickly touch on the aging clocks, because I think that's what a lot of people would point to, right? There are these epigenetic assays. So they look at changes on top of the DNA. So epigenetics really just means, changes that control gene expression without modifying your genome, so your DNA, but they can affect which genes get turned up or turned down. And methylation is the one that most people talk about. There are a bunch of other types of epigenetic changes, but that's the one most people look at. And people have developed these signatures of methylation that are correlated with chronological age. So these are the chronological age clocks. It turns out you can take a blood test and with a pretty high degree of precision, based on your epigenome, tell how old you are chronologically. That's not super useful because most of us know how old we are, but it works. And then kind of the next level is researchers have found you can look at different signatures in the epigenome that are predictive of future health outcomes. So future mortality risk 5 years, 10 years down the road, or future risk of developing age-related diseases. The hypothesis is that those are monitoring biological age. And so that's why you'll hear these things referred to as biological age clocks. I think the only thing I'll say about that, I'll say two things. One is we don't really know that they're measuring biological age. I think it's plausible they're measuring part of biological age, but there are a bunch of other ways you can measure biological age. You can probably do as well with facial imaging using artificial intelligence as you can with an epigenetic clock in terms of predicting future health outcomes or predicting chronological age. So I think if you have a sufficiently high dimensional data set, meaning you have lots and lots of features you can look at, and it's for a phenotype that changes characteristically with age, you can take a subset of the pieces of that data set or a subset of the dimensions and come up with a signature that will correlate strongly with chronological age. You can come up with a different subset that will correlate strongly with health or future health outcomes. That's what I think these clocks are measuring. So there's two things I would question now about the epigenetic clocks. I think they're super powerful research tools. I hope we get to a point where either alone or in combination with other biomarkers, they can be used as surrogate endpoints in clinical trials to assess whether an intervention is likely to work without having to do a full longevity study, which for obvious reasons is, not feasible in most clinical trials. I hope we get there. We're not there yet. I'm not to the point where I think that these things are useful for most consumers, even though there are lots and lots of companies selling these epigenetic tests. I kind of say they're for entertainment value only. And there's a couple of reasons why I'm not a big proponent of these tests for consumers. One is, like I said, I don't think we actually know what they're measuring. I don't think we can have a high degree of confidence that they are strongly predictive for future health outcomes, these specific tests, and we could talk about why I say that, or that they're predictive for whether or not an intervention, whether it's lifestyle or a drug or a supplement, is working at this point. That's one reason. The other reason is I think there is a unfortunate lack of transparency around a lot of these companies offering these tests. We really have no idea what the accuracy or precision of these tools are. It's probably different for different companies. We have no idea what their quality control looks like. There's just no regulation in this space. And so you kind of have to trust that these companies are doing it right. And I don't see a lot of reason to believe that. I'm not saying that they aren't, I just don't know. And so those two things combined make me very wary of, how much this has been sort of popularized in at least the biohacker community and to some extent beyond that. So the last thing I'll say is I'm a little bit wondering to what extent these tests are predictive of future health outcomes today. And the reason I say that is because the way these tests have been developed is they looked at bank samples from 10 years, 20 years ago. So these long-term longitudinal studies like NHANES or Baltimore Longitudinal Study of Aging have biobank samples from people. And they have enough people now who have gone on in the last 10 or 20 years to develop heart disease or cancer or die, that you can make correlations by looking at their samples from 20 years ago as to which signatures are associated with these health outcomes today. as we sort of alluded to earlier, the environment is fundamentally different today than it was 20 years ago. And so whether or not the same signatures are going to predict future health outcomes 10 years, 20 years from now as those that did 20 years ago, I think is an open question. They probably do to some extent, but my guess is that the accuracy is going to be less just because the human environment is different now than it was 20 years ago. So I think we'll have to wait and see.
That's super interesting. The one that I recently did was based upon the Dunedin Pace of Aging methylation profile.
Right.
I know that Horvath has one, Levine has one, and all that. But I think that particular study is quite interesting, that 40-year. But they also talk about, in those data sets, the facial imaging.
Right.
Do you use that in your clinical practice?
We don't. We don't. So we don't use the facial imaging. We do 3 different biological age tests from blood. So we look at the epigenome. So and it's worth saying. So you talked about the Dunedin Pace. So I should explain that briefly. So that is also an epigenetic signature. It's different than the sort of Horvath or Levine clocks in that signature is thought to predict the rate of aging. That's why it's called pace of aging, right? So these epigenetic biological age signatures were first correlated to biological age, right? And the idea is that your biological age can be different than your chronological age. The Dunedin Pace is correlated, at least in theory, to the rate at which your biological age is changing. And so it should be the case that particular signature is more responsive to fluctuations in the rate of aging, interventions, and can you move that in the right direction or not? And that's how a lot of people are interpreting it. So I do think those kinds of signatures, if they work as well as advertised, are really powerful, because then you can actually use those as surrogate endpoints for assessing whether or not an intervention is working, so to speak, for longevity. So that's the Dunedin Pace. And again, there's no reason why you couldn't create a similar pace of aging metric using other types of data. It's just a matter of having the right data set to do that. Okay. So what we do at Optispan right now is we are looking at 3 different aspects of biological age: epigenetics, blood glycans, which is another sort of protein measure in blood that changes with age, and a non-commercial test that looks at immune resilience and immune aging. So it looks at senescence of immune cells, T-cell activation, inhibition, and autophagy inhibition. We're using all of those as really sort of exploratory, though. We don't think that any one of those, or even all 3 of them combined, are necessarily more informative or even as informative as the other types of data that we collect. So it's really more about getting an overall picture of each individual from multiple different angles and then trying to synthesize that data. I think over time, we're also evaluating them. So I think over time, we expect that we will be able to start to make some, at least for our own purposes, conclusions about which tests are useful, maybe which aren't as useful.
Amazing.
Personally, I really like the immune test. I think I like it because the biochemical marks that are being measured are clearly important for immune function. And we know that immune function and immune resilience is really, really important as we get older. And so it's a combination of, in my view, both a molecular test and an immune functional test.
And so that will triangulate degree of senescent cell accumulation, amongst other things, as well as immune exhaustion. Is that what we're looking at?
So it's not that test is not a perfect measure of immune senescence, because we're really only looking at one protein marker of senescence, P16, but that's kind of the canonical protein marker of senescence. So we would expect it's at least strongly correlated with total immune senescence. Now, it's important to note that, immune senescence in a snapshot in time is really telling you only about the senescent burden at that moment in time. And that can change over time. It can change if you've recently had a severe COVID-19 infection, for example. So it's in and of itself only one metric that gives you one piece of information. But in general, if you look across the population, that marker goes up with age, and you can create predictions of immune age based on P16 and other markers in the immune system.
Going back to the COVID infection, that would increase the preponderance of senescent cells within your immune system, or the other way?
So I think I have to be careful here. So here's what I would say. I think it probably depends on when you look, right? So anytime your immune system is strongly challenged, you're going to go through, a series of sort of reactions to that challenge, which is going to include sort of massive expansion of the immune system to target that pathogen or that challenge and react to it. If it's in the case of something like a severe COVID-19 infection, what will often happen is you will have a prolonged hyperinflammatory response, right? That's the cytokine storm, which is counterproductive to say the least. I don't know for sure, but I would be surprised if you don't see a much higher burden of senescent cells in the immune system in that sort of a situation. But assuming everything resolves itself, you're going to go back to a steady state level, and presumably you're going to achieve something close to, if not close to, your baseline level of immune senescence. I think there are a variety of factors that can influence levels of P16 in this assay, and infection state is only one. What we're really trying to get at is less the sort of acute infection response and more the sort of baseline over time for an individual. How is that changing?
And then how would you characterize senescence or describe this to a high school biology student? What would you say, and why should people care about preventing the accumulation of these cells?
Yeah. So maybe one place to start is there are a variety of paths that cells in our body can go down when they stop doing their job. Okay. So all of our cells in our body have a job to do, but sometimes they stop working the way they're supposed to. So one path is apoptosis, which is just cell death, right? So the cells die, they go away. That might seem like it's a problem, but it's really not, because our stem cells can replenish and regenerate. So you can have cells die, or you can have cells that refuse to die. They just stop doing their job. Those are senescent cells. And we know that senescent cells are important in a few specific cases. So there are cases of programmed senescence, like during early fetal development, where we need these cells to kind of stick around. They give off signals that are important for continued development. Senescent cells also seem to be important in wound healing for the same reason. So right around the site of a wound, you will have these cells that stop doing their job. And what I mean by that really is they stop dividing, they stop functioning metabolically like they normally would. They go into this non-dividing, arrested state, and then they start to give off signals, mostly inflammatory signals. Okay. But as we get older, we also have senescent cells accumulate. And cellular senescence in the context of aging in particular is really important because it's a potent anti-cancer mechanism. Because the other thing that can happen, the other path cells can go down, is they become cancer. Nobody wants that. So dying is probably best in most cases. Becoming senescent is better than becoming cancer. And so, as we get older, our bodies accumulate these cells that just kind of hang out. Our immune system is pretty good at clearing them when we're young, but the immune system gets less able to clear the senescent cells as we get older. So it wouldn't be a problem if they just hung out. But they don't just hang out. They're kind of like rabble rousers. So they're like guys who on the work site who stop working, but then they're off getting other people to stop working and start goofing off, right? So they give off all these chronic inflammatory signals, which then have an impact on the cells around them, can promote tumorigenesis in surrounding cells, or can promote other cells throughout the body to become senescent, or can lead to high-grade chronic inflammation, which is what many of us experience as we get older. So the thought is, if we could get rid of the senescent cells, that would be a good thing, because we don't want them around. The immune system does it when we're young, but it's not able to do it as we get older. And so people have tried to develop these drugs called senolytics or other strategies to kill specifically senescent cells. So that's the concept behind this idea of senolytics. You have a drug, it's going to specifically target senescent cells, kill senescent cells, not do anything to the surrounding cells. In theory, that would be great. In reality, no drug is that clean. And I'd say the current crop of senescent cells are mixed at best. So they do kill some fraction of senescent cells. They also do bad things to cells that aren't senescent. And so people are really trying to work on better and better second generation, third generation senolytics at this point. What we know is that if you treat mice with senolytics, you can increase lifespan. You can improve multiple measures of health span. There are some early indications from, again, underpowered clinical trials in people that you can get some benefits. I think idiopathic pulmonary fibrosis is a place where a lot of attention has gone. There seem to be a lot of senescent cells there. So that's one place people are looking. Some eye disorders as well. There's a lot of senescence. And so people are trying to think about where can we go? What disease conditions can we look at where there's a high burden of senescent cells to try to demonstrate some clinical efficacy? But my sort of read is that the current crop of senolytics isn't quite there yet. But in theory, if you could develop very specific senolytics, almost certainly there would be some clinical benefit. It's also probably worth noting that rapamycin is not a true senolytic. It is an indirect senolytic in the sense that it can actually, in aged animals, boost the immune system and the immune system's ability to clear senescent cells. It is also what's called a senomorphic, meaning it is pretty good at turning down the bad signals that senescent cells are giving off. So if you look at these inflammatory cytokines that senescent cells secrete, if you treat senescent cells with rapamycin, or if you give old mice rapamycin, you see that it is pretty potent at turning that down. So it does seem to be some overlap between rapamycin and senolytics in terms of mechanism of action.
Amazing. And outside of rapamycin, what other cocktails are you excited about? There's quercetin, dasatinib, there's all these different things. Is there anything that has more or less efficacy from what you've read?
Yeah, I mean, I would say dasatinib plus quercetin is probably the one that has the most evidence supporting it at this point in terms of small molecules. There are genetic constructs in mice where the mice are engineered in a way that if you feed them a certain drug, the senescent cells will commit suicide. That works really well in mice. That's not relevant for people. But I think right now DNQ is probably the one where there's the most data. There are a bunch of other molecules that people will say have senolytic activity. Most of those, it's pretty unclear at the doses that could be achieved by just supplementing with those molecules if they really have any potent senolytic activity or not.
And what about exercise?
Yeah, so exercise is interesting. I don't know that exercise itself has a direct senolytic activity. I think it's very likely, and there's good evidence to support the idea that regular exercise can have an immune protecting, immune boosting effect. So you might expect an indirect benefit from exercise in terms of allowing the immune system to be better able to clear senescent cells. And that's going to be true for cancer surveillance as well. I think this is, again, something that maybe isn't as well appreciated as it should be. Our immune system is probably our most potent anti-cancer mechanism directly, right? We are constantly developing early stage cancers that get cleared by our immune system. As a consequence of chronic inflammation, that leads to an inability of the immune system to respond to normal pathogens or cancer surveillance. So we see a decline in cancer surveillance. We see a decline in senescent cell surveillance. That decline in senescent cell surveillance creates this vicious cycle where there's now more inflammation, which is further causing decline. So I think things like exercise, this is a place where you could make a case for fasting, right? Where fasting, because of its anti-inflammatory effects, may have a beneficial effect on immune function and senescent cell clearance, things like that. Again, if you were to make up for fasting by overeating, you might offset those benefits, but it's plausible that fasting could have some benefits through that mechanism.
Interesting. So exercise by way of immune enhancement could lead to the favorable changes in purging the cells. It's not directly senolytic.
Again, there may be cases where it's directly senolytic, but I think that it's more plausible just from a mechanistic perspective to speculate about effects on immune system and then senescent cell clearance. Again, it's also probably worth saying, I think the way people measure senescence is tricky, and I still don't know that there is full consensus in the field about how you would actually measure, even in a mouse, like what is the senescent cell burden? What is the gold standard way to do that? There are a whole bunch of markers that people use. So you could probably find a paper that says that exercise is senolytic. You'd really have to look at what they measured and did they actually measure senescent cells and how believable is it? This is a challenge in general, though, with the senescence field, that because there isn't consensus in what people measure, oftentimes people will default to the lowest common denominator, make claims about senolytics, but when you actually look at the data, it's like, that probably isn't really a senolytic.
It seems that obesity and the adipocytes themselves are particularly problematic.
Yep.
Would you agree with that?
Yeah. I mean, again, I think the preponderance of evidence at this point is that accumulation of senescent cells in fat, particular visceral adipose, is driving a lot of chronic inflammation, age-related inflammation. That has a big impact on metabolic dysfunction. Again, it's sort of this vicious cycle, right? You get metabolic dysfunction, you get less immune function, you get less clearance of senescent cells. But yeah, I mean, I think that is pretty clear that at least right now, probably if you had to pick one tissue that you could clear senescent cells in, fat would be the place that you would start.
Yeah. And speaking of another tissue to go, I want to go back to rapamycin and the dosing. We talked about ovarian senescence.
Yeah.
Now people know what senescence is and why that could be problematic in the context of the ovaries, particularly if one wants to have children. But we wanted to kind of close the loop on the dosing of rapamycin, because I think, again, Alan Green has been recommending, I think you mentioned 6 milligrams, once a week.
Yeah.
Morning, night, body weight dependent. Where are people landing with that?
Yeah. So I can tell you what most people are doing. I unfortunately can't tell you what's optimal because we just don't know. We just don't have the data. So from the double-blind, randomized, placebo-controlled clinical trials. Let's start with the gold standard data. We know from studies that Joan Manick did, several years ago now with everolimus, which is a derivative of rapamycin, that once weekly dosing around the 5 milligrams per week dose gave positive effects on boosting flu vaccine response in the aged immune system and had side effects that were really no different than placebo. So most people have gravitated towards that once weekly dosing around the 6 milligrams per week for off-label use of rapamycin. There are some speculative but plausible biochemical reasons why once weekly dosing could make sense. The one most people will point to is that, and this gets in the weeds just a little bit, but I won't go too far in the weeds. There are 2 complexes that mTOR acts in. mTOR complex 1 or mTORC1 and mTOR complex 2, mTORC2. mTOR complex 1 is the rapamycin sensitive complex. So biochemically, rapamycin is a specific inhibitor of mTOR complex 1. If you have chronic inhibition of mTOR complex 1, you will get off-target effects on mTOR complex 2. The idea, I was going to say, I don't want to say I'm not completely convinced this is the case, but some people are. The model that's been put out there is that the positive effects of rapamycin for longevity and health are predominantly, if not exclusively, due to inhibition of mTOR complex 1, and the side effects are predominantly, if not exclusively, due to mTOR complex 2. The reason why I'm going down this tangent is that weekly dosing, because you don't ever get that chronic inhibition of mTOR complex 1, probably doesn't lead to the off-target effects on mTOR complex 2. So it's probably a more specific mTOR complex 1 inhibitor in that sort of context. And so that's one biochemical rationale for this weekly dosing. And that does sort of fit the side effect data I talked about from Joan. So most people are doing 6 milligrams once a week, but there's a big variation. So some people are doing 1 milligram once a week. Some people are doing 20 milligrams once a week. We don't have any data on efficacy one way or the other. Again, my intuition is that 1 milligram once a week is probably too low to have any real potent anti-inflammatory effects. And again, if we look at Joan's study, which is really the only data I'm aware of to go on at this point, 20 milligrams once a week may not be past the optimum for efficacy, but it's probably getting to the point where you start to uncover more side effects. Mm-hmm. So intuitively, somewhere around 6 to 10 milligrams once a week, I think is a reasonable place. And again, that's where most people are landing.
Mm. And what were you dabbling with when you do a cycle periodically? And your anecdotal story about your frozen shoulder is pretty impressive. Yeah.
So that was at 6 milligrams once a week. Okay. I've gone between 6 and 10. I did try 20 once just to see it. So to be honest with you, I've noticed no differences between 6 and 10. Or even at 20. So no acute effects that I would ascribe to dose differences. I do think, and again, we don't have a lot of data, but it seems certainly reasonable that certainly there will be different tissue of it bioavailability. No question. We know that for rapamycin. And so at different doses, you're going to get different degrees of mTOR inhibition in different tissues. And so if you're, and again, this is very speculative because we don't know for certain that mTOR is going to prevent dementia or rapamycin is going to prevent dementia. But in mice, it does, okay? So if your goal is you're taking rapamycin because you, say, have 2 ApoE4 alleles, you're at high risk of dementia, you might want to take a higher dose with the rationale that rapamycin doesn't get across the blood-brain barrier very efficiently. You want to get higher brain penetration. That's reasonable. So I think it depends on the context and what the hoped for outcome is. You can sort of be guided in terms of dosing there. The other thing that this is probably just worth saying is because rapamycin is metabolized by the cytochrome P450 system, it is inhibited by grapefruit juice. So some people will take grapefruit juice with their rapamycin, and you do get about, I think it's like 3 to 6-fold higher peak levels in the blood from rapamycin dosing.
So in real time, you do concomitantly grapefruit juice with the rapamycin.
Right.
Interesting.
Right.
And so you mentioned the ApoE4 genotype.
I mean, I don't. Some people do.
Some people do. Sure, that makes sense. The genotype having 1 or 2 alleles of the ApoE4, would that be the best clinical use case for a prophylactic regime? Do you think those people are most likely to experience the greatest benefit?
It's a good question. I don't know if it's the best use case. It's a reasonable use case, right? Again, and this is purely my observation, right? I don't have any quantitative data to back it up. It is my perception among all the people who I've talked to who've used rapamycin. I've talked to a lot of people who've used rapamycin off-label. There are a subset of people that feel nothing, or they get mouth sores. Some people will get mouth sores. That's about the only real side effect that I've picked up on. Some people will feel nothing. I'd say that's most people, maybe 2/3. They just don't, maybe not. A lot of them keep taking it, but they don't really notice anything obvious. And then there's a set of people where they're like, oh my God, I feel so much better. And I think those are the people who have very high levels of chronic inflammation. So like my case with my frozen shoulder, right? It was a nightmare. I couldn't sleep. I couldn't throw a ball with my kid. 10 weeks later, I was back to normal, right? Wow. So I think the people who have that sort of experience are the people who have very high levels of chronic inflammation, and it's influencing a whole bunch. It's influencing how they feel, right? And so I think there are use cases like that where it's an acute condition, like with my frozen shoulder. Again, I wish somebody had done a trial by now, right? To actually demonstrate to how many people who have frozen shoulder, a lot of people do, how frequently does it actually have a positive benefit, right? But for people like that, clearly there's a huge improvement in quality of life. That's a use case. We don't know yet about periodontal disease, body composition, ovarian failure, right? We'll wait and see how the data plays out. Those could all be very reasonable use cases as well. And then there are a bunch of people who have clinical autoimmunity, right? And those are the places where there are case reports in the literature for different autoimmune conditions where rapamycin seems to be effective. That again makes sense. To some extent, chronic inflammation with age is an autoimmune disorder. Aging to some extent is a low-grade autoimmune disorder for many people.
Yeah. Even obesity, I mean, it meets 4 of the 5 defining criteria of autoimmunity, which is really interesting. So, I think we love to talk about body fat and mental performance, but I think a big theme that you've highlighted here, especially when it comes to senescence, is the health of your immune system really dictates how you will age. I mean,
that's absolutely right.
So,
and I do want to say, I mean, anytime I'm in a conversation, we tend to go down the rapamycin trail, which is great. I love talking about rapamycin, but you can have positive impacts, as we've already alluded to, on chronic inflammation through diet, right? Through exercise, through cutting out alcohol, right? All of those things are probably more potent than rapamycin, or at least equally potent. I mean, rapamycin, again, is a very specific drug with one biochemical target. Mm-hmm. Those other interventions are non-specific and they do a lot, but they're also potently anti-inflammatory. And so I certainly wouldn't want to suggest that people should take rapamycin and not worry about nutrition or exercise or sleep. I think for some people, it can be a potentially valuable addition to those pillars.
I agree 100%. Was there any concomitant rollout of the COVID-19 vaccines with rapamycin that you knew of?
No, and this is sort of a sad story. So Joan was chief science officer at a company called Restore Bio in in 2019. And they were moving forward. So they'd gotten the rights to the mTOR inhibitors from Novartis. So they were moving forward based on Joan's earlier work with phase 3 clinical trial with an mTOR inhibitor. It's called RTB101, not a rapamycin derivative. We can come back to that in a minute, where they were actually testing whether or not RTB101, this mTOR inhibitor, could boost flu vaccine response in healthy older people. They were doing this in 2 phases, one in the southern hemisphere, one in the northern hemisphere, because the flu seasons are opposite. They'd gotten halfway through. I remember this timing because I was with Joan at the Gerontological Society of America meeting, which is always in November. It's November 2019. Gotten halfway through the clinical trial, and they did an interim analysis and were not hitting their endpoint. Okay, so the endpoint here is important. We'll come back to that. They weren't hitting their endpoint, so the board of directors decided to shut the trial down in November 2019. Of course, we all know what happened in April 2020, right? Joan went back and did a post hoc analysis on the data from that study. And it turns out the people who got RTB101, or looking at the prior studies, the people who'd gotten the rapamycin derivative, were protected against at least 3 different viral families. I don't remember what one of them was. One of them was influenza, pretty big deal. The other one was coronavirus. Of course, it wasn't COVID-19 because nobody knew about COVID-19 in 2019 when these patients were dosed. So I think this is a very unfortunate situation where, because of the endpoint, which again, I'll come back to, and because the board of directors, basically chickened out, they cut the trial off too soon. If they'd known what was coming, I suspect they might have made a different decision. And boy, wouldn't it have been nice if we had a drug that older people could take in 2020 to prevent or reduce the severity of COVID-19 infection. Okay, I know it's a little bit in the weeds, but the endpoint here is it's important to appreciate why clinical trials fail. Clinical trials can fail for all sorts of reasons. You have to make all sorts of decisions when you're designing a clinical trial. Some of them are based on science, some of them are based on pragmatism. How much money do you have? What's your endpoint? So in the first 2 clinical trials where everolimus or everolimus plus RTB101, this other mTOR inhibitor, boosted vaccine response, the endpoint was clinically validated infections. So in other words, a PCR test for the virus. For reasons I don't really understand, FDA going into the phase 3 made them change the endpoint to patient-reported infections, which makes no sense from a scientific perspective. And people think they're sick all the time when they're not sick, or vice versa. But that was what the endpoint was, and that was the endpoint they were failing to hit when they got halfway through the clinical trial. So I think it was just a combination of unfortunate timing, unfortunate, luck in the way that the trial was designed. I also did say RTB101 is not a rapamycin derivative. They took the rapamycin derivative out of that trial, again, for reasons that you'll get a different answer depending on who you ask. I speculate at least part of it was based on IP. Yeah. They had a longer patent life on the RTB101, which works by a different mechanism. So it still inhibits mTOR, but it also has off-target effects, and it works by a different mechanism. So might it have hit the endpoint if they had the rapamycin derivative in there? We'll never know because that trial wasn't done, but it had in prior trials.
Maybe suggesting that the whole is greater than the sum of the parts. Like there's something unique about a rapamycin derivative, the yeast, isn't it? Or
so rapamycin. Maybe it's worth just briefly talking about the way rapamycin works. So rapamycin inhibits mTOR, but it does it through what's called an allosteric mechanism, meaning it doesn't directly bind to the active site of mTOR. Active site is where the enzyme does its job. That's where the action happens, right? So rapamycin binds another protein called FKBP12. That complex then goes and breaks up mTOR complex 1. So it's an allosteric indirect inhibitor of mTOR. There are another class of drugs that are called ATP competitive inhibitors that bind the active site. They look something like ATP, which gets into the active site. And so they get into the active site and basically inhibit the enzyme that way. So it's a fundamentally different mechanism of action. The thing about the ATP competitive inhibitors is they also tend to hit other proteins in the same family, other kinases, because many kinases have a similar structure to their active site. And so RTB101 is a class of drugs called a dual kinase inhibitor because it also hits PI3 kinases, which have all sorts of, implications in cancer and growth. So it could be that it's because RTB101 is a little bit less specific. It could be that there's something about the mechanism of action and the way it inhibits mTOR complex 1 and not mTOR complex 2. That's certainly different about rapamycin. So I just think we don't know, but they do seem to be potentially different in the effect they have on the aged immune system. And yeah, in the phase 2 clinical trial where they combined both everolimus, which is the rapamycin derivative, and RTB101, they did see better effects than everolimus alone.
Interesting stuff. We could go all day with all these different topics. Matt, really appreciate you coming on the show. If folks want to connect with your podcast and your work, how do they do so?
Yeah, so our podcast is called the Optispan Podcast. We're on YouTube and Apple and Spotify, and pretty easy to find me. My handle on X is @mKaeberlein, and I hope people tune in.
Amazing. Thank you for coming on.
Yeah, thanks a lot.